The Fluid Mosaic Model is a widely accepted model that describes the structure of cell membranes proposed by Singer and Nicholson in 1972. It revolutionized our understanding of cell membranes. The model suggests that the lipid bilayer of the cell membrane behaves like a fluid. Phospholipids, which are the primary constituents of the lipid bilayer, have a hydrophilic water-attracting head and hydrophobic water-repelling tails. These molecules are able to move laterally within the plane of the membrane. This lateral movement gives the membrane its fluidity, allowing various molecules, such as proteins, to move within it. The mosaic part of the model refers to the diverse array of proteins that are embedded within or attached to the lipid bilayer. These proteins have various functions, including transport of molecules across the membrane, cell recognition, signaling, and structural support. They are interspersed throughout the lipid bilayer in a mosaic-like pattern. The basic structure of the cell membrane is a lipid bilayer composed primarily of phospholipids. These phospholipids have a hydrophilic phosphate head and hydrophobic fatty acid tails. In the aqueous environment of the cell, these phospholipids arrange themselves into a bilayer with the hydrophobic tails facing inward and the hydrophilic heads facing outward, interacting with the surrounding water. Integral proteins span the entire lipid bilayer, with portions extending into both the cytoplasmic and extracellular environments. These integral proteins can function as channels, carriers, receptors, or enzymes. Peripheral proteins are attached to the surface of the membrane, often associated with the cytoplasmic sign, and may serve as enzymes or play a role in cell signaling. Cholesterol molecules are interspersed within the lipid bilayer. They help to regulate the fluidity and flexibility of the membrane. Cholesterol molecules can interact with the fatty acid tails of the phospholipids, reducing the mobility of the lipid molecules and thus affecting the overall fluidity. Some proteins on the extracellular surface of the membrane have attached carbohydrate chains. These carbohydrate chains can serve as recognition sites for other cells or molecules, playing a key role in cell-cell recognition and communication.